Hello students and welcome to the lecture on Patterns of Inheritance for Monogenic Traits. The main objectives of today's lecture are to give a general idea of monogenic inheritance, to explain dominant and recessive autosomal inheritance, to explain sex-linked inheritance, both X and Y-linked, to explain sex-limited and sex-influenced traits, to explain mitochondrial inheritance. To begin with, let me first of all give you a very brief introduction about the topic. Let us begin with the definition of monogenic inheritance. Monogenic inheritance refers to the kind of inheritance whereby a trait is determined by the expression of a single gene or allele, not by several genes as in polygenic inheritance. It is the inheritance of characteristics that are determined by a single gene pair. These characteristics showing discontinuous variation, that is, those represented by two or more contrasting characteristics. There are few easily visible normal external characteristics that show monogenic inheritance. Examples are eye color, large or small ear lobes, sticky or dry earwax, ability or inability to curl the tongue, ability or inability to taste the substance, ability or inability to hyperextend the thumb, that is, hitchhiker's thumb. However, there are numerous examples of diseases that follow monogenic pattern of inheritance. Inherited genetic diseases are examples of discontinuous variation. The disease is either present or is not, and there is no intermediate range. They show monogenic inheritance. Now, let us start with the first part of our topic, that is the autosomal dominant inheritance. First, I shall give you the pattern of inheritance followed by autosomal dominant trait. Autosomal dominant inheritance manifests itself in the heterozygote. The trait is equally represented in males and females. The trait shows vertical inheritance through generations, that is, transmission from parent to offspring. Affected parents have 50% chance that their offspring will be similarly affected. The trait does not skip generations. It's transmitted by either sex. Now, let me give you some examples of autosomal dominant disorders. First, achondroplasia. It's a type of dwarfism characterized by short limbs, a normal sized trunk, and various skeletal abnormalities. Second, adult polycystic kidney disease. Multiple cysts in the kidneys and liver causing symptoms and complications in adult life and leading to progressive renal failure. Third one, brachydactyly, meaning short fingers or toes, usually the middle phalanx is short. Congenital spherocytosis. The red blood cells are in the form of spheres rather than flat biconcave discs. They cause anemia. Familial adenomatous polyposis. Numerous polyps in the large intestine becoming cancerous in most cases. Familial hyperlipidemia. Elevated blood levels of low density lipoproteins predisposing to coronary heart disease at a very young age. This is the commonest autosomal dominant disorder. Next, Huntington's disease. It is a late onset neurological disorder appearing usually around the age of 35 years and characterized by abnormal involuntary breathing movements, chorea, and behavior and personality disorders. Marfan syndrome. A disorder of connective tissue. It causes abnormalities of the skeleton, including tall stature, wide arm span, spidery fingers, and deformities of the sternum, cardiovascular abnormalities such as aortic aneurysm or heart valve incompetence and subluxation of the lenses. Neurofibromatosis, characterized by numerous caveolate pigmented patches and multiple benign tumors of the nerve sheaths, neurofibromas, causing irregular swellings in the skin. Postaxial polydactyly, characterized by an extra digit on the side of the little finger or little toe. This may vary from a small tag to a well-developed digit. Tuberous sclerosis, that is adenoma sebaceum. It's characterized by an adenomatous rash on the cheeks, calcification in the brain, and fits. However, some autosomal dominant disorders do not always appear to follow the general rules stated above. Some cases do not have the characteristic pedigree and have a negative family history. This may be due to one of the following phenomena. First one, conditions that manifest themselves late in life. The typical example is Huntington's disease. Although the gene for Huntington's disease is present from the time of conception, affected individuals are normal until the symptoms appear, usually around the age of 45 years. Many individuals who carry the abnormal gene marry and have children before the disease appears. The age at onset of the disease is very variable, ranging from 20 to 70 years. Some individuals who have the abnormal gene might die from unrelated causes before the disease appears. Thus, the pedigree may show an unaffected person who has affected offspring. Second, new mutations. Autosomal dominant conditions that have a negative family history are usually the result of new mutations. Individuals with a severe disorder such as achondroplasia often have reduced chances of getting married and have children because of medical or social reasons. Achondroplastic dwarfs have 50% of their children similarly affected. Most cases of achondroplasia, however, have normal parents. These cases are the result of new mutations. For a normal couple who had a child with achondroplasia arising as a new mutation, there would be no increased recurrence risk for their other children. However, the affected child would have a 50% chance of having affected offspring. Third, variable expressivity. There is often a considerable variation in the severity of the disorder or in the type of abnormalities present, although the genetic defect is the same. This is referred to as variable expressivity. Most genetic diseases show some degree of variable expressivity. For example, in postaxial polydactyly, the extra digit may vary from a small skin tag to a fully formed digit. In Marfan syndrome, affected individuals often do not have all the characteristics. One individual may be present with tall stature and subluxated lenses, another with an aortic aneurysm. Sometimes the variability may be so great that there may be little resemblance in phenotypic manifestations of an affected individual and his affected offspring. For example, in one type of limb reduction deformity, the disorder may be expressed to variable degrees from a missing finger to a missing leg. Fourth one, penetrance. In some cases, individuals who carry an abnormal gene do not appear to manifest any symptoms of the disease, although their offspring may be affected and it would appear in the pedigree that the disease has skipped a generation. The reason for this could be reduced penetrance. Penetrance is a measure of how frequently a gene is expressed. Some autosomal dominant conditions, example achondroplasia, have 100% penetrance. That is, when the gene is present, the disease always expresses itself. In other conditions, there may be reduced penetrance. Retinoblastoma is an autosomal dominant condition that has 85% penetrance. That is, only 85% of persons carrying the abnormal gene show symptoms of the condition. The remaining 15% never developed retinoblastoma but can still transmit the gene to their offspring. Now, moving to second part of our topic, the autosomal recessive conditions. Let us again first discuss the pattern of inheritance of this condition. The condition manifests itself only in homozygotes. The parents of an affected individual are both heterozygous carriers but are phenotypically normal. The condition is often present among SIBs but is not usually transmitted through generations. The offspring of heterozygous parents have 1 in 4 chance of being affected. The ratio of affected to normal offspring is 1:3. An almost equal number of affected males and females. In case both the parents are affected, all children would be affected. Now, let me give you a few examples of autosomal recessive disease. Number 1 is albinism. There is lack of pigment in the skin, hair, and eyes due to a deficiency in one of the enzyme, example tyrosinase, that is necessary for formation of melanin pigment from tyrosine. Lack of pigment may cause visual problems and sensitivity of the skin to light. Number 2, beta thalassemia. The adult type of hemoglobin, that is HbA, is not produced or is produced in very small amounts due to a defect in the production of beta globin. This causes severe anemia beginning in early childhood and requiring frequent blood transfusions. The bone marrow increases greatly in amount, trying to compensate for the anemia, causing a thickening of the bones of the skull and face. Congenital hypothyroidism. This is due to lack of secretion of hormone thyroxine. Cystic fibrosis, a disorder in the secretions of gland affecting mainly the respiratory system, pancreas, and sweat glands. Death in infancy may result from recurrent severe pulmonary infections. Galactosemia. This is due to lack of an enzyme required to metabolize galactose. High levels of galactose are present, causing mental retardation, cataracts, and cirrhosis of the liver. Gangliosidosis. Deficiency of the enzyme beta-galactosidase causes accumulation of the ganglioside in the tissue, including the brain, bone marrow, liver, and spleen. Involvement of the brain causes fits, severe mental deterioration. Phenylketonuria, a defect in phenylalanine metabolism causing mental retardation unless a special diet is started early enough in infancy. Sickle cell anemia, a disorder of the hemoglobin molecule causing the red blood cells to become sickle-shaped and rupture under conditions of low oxygen tension. This results in severe hemolytic anemia. Now, let us discuss the third part of today's topic, that is sex-linked inheritance. The sex chromosomes X and Y, besides carrying the sex-determinant genes, also bear genes for several other characters. These additional genes travel along with the sex-controlling genes from generation to generation and are therefore called sex-linked genes. The characters controlled by the sex-linked genes are termed sex-linked characters. Inheritance of sex-linked genes or traits is known as sex-linked inheritance. Their location in the sex chromosome is called sex linkage. Most sex-linked genes are located on X chromosome, forming X linkage. A few genes occur on the Y chromosome, forming Y linkage. The Y linked traits are transmitted only through the male. For example, gene for sex determination in mammals. The Y chromosome carries so few genes that it is described as genetically inert or genetically empty. The sex linkage was first discovered by Morgan in 1910 in the fruit fly Drosophila melanogaster. This fly has XX and XY sex chromosomes in the female and male respectively. Its eye color gene is located in the X chromosome and there is no corresponding allele in the Y chromosome. The male expresses a sex-linked recessive trait even if it has a single gene for it, whereas the female expresses such a trait only if it has two for it. The normal eye color is red and is dominant over the mutant white eye color. The following crosses illustrate the inheritance of X-linked eye color in Drosophila. Red-eyed female cross with white-eyed male. If a homozygous red-eyed female fly is mated with a hemizygous, that is, having a single allele for a trait, white-eyed male fly, all the F1 flies, irrespective of their sex, are red-eyed. When the red-eyed male and female flies of F1 are intercrossed, the F2 flies are in the ratio of 2 red-eyed females to 1 red-eyed male to 1 white-eyed male. Thus, the red-eyed and white-eyed flies are in the ratio of 3:1 in F2 generation, that is Mendelian monohybrid ratio. The above cross shows that a recessive X-linked trait follows crisscross inheritance, that is, transmission from the father to the grandsons through the daughters. The latter are called carriers because they have a trait but do not express it. Now, white-eyed female cross with red-eyed male. In this cross, a homozygous white-eyed female fly is mated with a hemizygous red-eyed male fly. This cross is reciprocal of the previous cross and should yield the same result. But in its F1 progeny, all the males are white-eyed and all the females are red-eyed. This result shows that eye color and sex are linked. When a red-eyed female fly and a white-eyed male fly of F1 are intercrossed, F2 progeny comprises approximately equal number of red-eyed females, white-eyed females, red-eyed males and white-eyed males. The above cross shows that the father transmits its dominant sex-linked trait, that is red eyes, to his grandsons as well as granddaughters through daughters. This proves that the eye color gene lies in the X chromosome. The sex-linked inheritance pattern can be studied under the following three headings: patterns of X-linked recessive inheritance, patterns of X-linked dominant inheritance, patterns of Y-linked inheritance. First, patterns of X-linked recessive inheritance. Most affected individuals are males. Usually, none of the offspring of the affected male will be affected, but all his daughters will carry the gene in masked heterozygous condition. None of the sons of an affected male will be affected. Affected males result from mothers who are affected or who are known to be carriers, that is heterozygotes. Affected females come from affected fathers and affected or carrier mother. The sons of affected females should be affected. Approximately half the sons of carrier mother should be affected. Now, let me give examples of X-linked recessive inheritance disorders. Number 1 is the Duchenne muscular dystrophy. It occurs due to the deficiency of protein dystrophin. Dystrophin is a protein of high molecular weight that is associated with the transmembrane glycoprotein complex of skeletal muscle cells. It is characterized by muscular degeneration and weakness, and sometimes also associated with mental retardation. Hemophilia A. It's a classical form of blood clotting deficiency resulting due to the deficiency of clotting factor 8. Hemophilia B, known as Christmas disease, it results due to deficiency of clotting factor 9. Glucose 6-phosphate dehydrogenase deficiency. Deficiency of glucose 6-phosphate dehydrogenase enzyme causes severe anemic reactions following the intake of primaquines in the drugs and certain foods including fua beans. Leishman syndrome. Deficiency of hypoxanthine guanine phosphate ribosyl transferase enzyme, that is AGPRT. It leads to motor and mental retardation, self-mutilation and early death. Color blindness, proton type. It is due to the insensitivity to red light. Color blindness, deuterium type, due to insensitivity to green light. Now, patterns of X-linked dominant inheritance. It's frequently found in females than in males. Affected females come from affected mothers or fathers. Affected males come from affected mothers. Approximately half of the children of an affected heterozygote female are affected. All daughters, but none of the sons of an affected father are affected. The trait usually does not skip generations. Example of X-linked dominant inheritance: hypophosphatemia, vitamin D resistant rickets, heredity enamel hypoplasia. The third X-linked pattern is the Y-linked inheritance. Genes in the non-homologous region of Y chromosome pass directly from male to male and show following pattern of inheritance. Affects only males. Affected males always have an affected father. All sons of an affected male are affected. Example: Ichthyosis hystrix gravis hypertrichosis. The next part of the topic is sex-limited inheritance. As we know, certain genes produce characters in one sex only, but not in both even though they are present in both the sexes. Such characters are called sex-limited characters. The genes of these characters are located in the autosomes instead of in the sex chromosomes. So, the sex-limited traits are the autosomal traits in which the dominant expression depends on the sex hormones of the individuals. The sex-limited characters develop only in the presence of sex hormones. So, the sex-limited genes are genes that are present in both sexes of sexually reproducing species but are expressed in only one sex and remain turned off in the other. In other words, sex-limited genes cause the two sexes to show different traits or phenotypes despite having the same genotype. This term is restricted to autosomal traits and should not be confused with sex-linked characteristics, which have to do with genetic differences on the sex chromosomes. Sex-limited genes are also distinguished from sex-influenced genes, where the same gene will show differential expression in each sex. Sex-limited genes are responsible for sexual dimorphism, which is the phenotypic difference between males and females of the same species. These differences can be reflected in size, color, behavior, for example, levels of aggression and morphology. An example of sex-limited genes are genes which instruct the male elephant seals to grow big and fight, and at the same time instructing female seals to grow small and avoid fights. These genes are also responsible for some female beetles' inability to grow exaggerated mandibles. Now, examples of sex-limited inheritance. Bright plumage. Bright plumage in some male birds. Milk secretion in female mammals. The bulls have the genes for the milk production which they transmit their daughters, but they or their sons are unable to express this trait. The production of milk is therefore limited to variable expression only in the female sex. Beard. Beard development in human beings is a sex-limited trait, as men normally have beards whereas women normally do not. Likewise, the genes for male vice, body hair and physique are autosomal in human beings, but they are expressed only in the presence of androgens, which are absent in females. Now, moving to the next part of today's lecture, that is the sex-influenced inheritance. In sex-influenced traits, an allele is dominant in one gender but recessive in the other gender. In such type of inheritance, the dominance is influenced by the sex of the bearer. Thus, male and female may be similar for a particular trait but give different phenotypic expression of the same trait. This may be clear with few examples. Hair lip. Hair lip is more common and more severe in males than in females. Pattern baldness in man is also a sex-influenced trait. In pattern baldness, hair is lost on the head top but persists as a fringe low on the head. It is more common in males than in females. Women who have genotype for pattern baldness typically show only thinning of hair. Moreover, man becomes bald if he has only one allele for baldness, whereas the woman needs two alleles for baldness to lose hair. This is the reason why bald women are rare. Now, let us discuss the last part of today's lecture, that is the mitochondrial inheritance. Mitochondrial genes show maternal inheritance because all the mitochondria a zygote has come from the cytoplasm of the ovum. Thus, in multicellular organisms, mitochondrial DNA is inherited from the mother. This pattern of inheritance is also known as extrachromosomal inheritance. Mutations in the mitochondrial genes cause some rare human disorders. For example, a person with a disease named mitochondrial myopathy suffers weakness, intolerance of exercise, and muscle deterioration. Let us discuss few patterns of mitochondrial inheritance. Number 1 is the female inheritance. Number 2, mitochondrial bottleneck. Number 3, the male inheritance. And the number 4th, three-parent inheritance. The first one is female inheritance. In sexual reproduction, mitochondria are normally inherited exclusively from the mother. The mitochondria in mammalian sperms are usually destroyed by the egg cell after fertilization. Also, most mitochondria are present at the base of the sperm's tail. Sometimes the tail is lost during fertilization. The fact that mitochondrial DNA is maternally inherited enables genealogical researchers to trace maternal lineage far back in time. Biologists can determine and then compare mitochondrial DNA sequences among different species and use the comparisons to build an evolutionary tree for the species examined. Another pattern of mitochondrial inheritance is the mitochondrial bottleneck. Entities undergoing uniparental inheritance and with little to no recombination may be expected to be subject to Muller-Schreier, the inexorable accumulation of deleterious mutations until functionality is lost. Animal populations of mitochondria avoid this build-up through a developmental process known as the mitochondrial DNA bottleneck. The bottleneck exploits stochastic processes in the cell to increase in the cell-to-cell variability in mutant load as an organism develops. A single egg cell with some proportion of mutant mitochondrial DNA thus produces an embryo where different cells have different mutant loads. Cell-level selection may then act to remove those cells with more mutant mitochondrial DNA, leading to a stabilization or reduction in mutant load between generations. The mechanism underlying the bottleneck is debated, with the recent mathematical and experimental meta-study providing evidence for a combination of random partitioning of mitochondrial DNAs at cell divisions and random turnover of mitochondrial DNA molecules within the cell. The male inheritance. Doubly uniparental inheritance of mitochondrial DNA is observed in bivalve mollusks. In those species, females have only one type of mitochondrial DNA, whereas males have F-type mitochondrial DNA in their somatic cells, but M-type of mitochondrial DNA, which can be as much as 30% divergent in germline cells. Paternally inherited mitochondria have additionally been reported in some insects such as fruit flies, honeybees, and periodical cicadas. Male mitochondrial inheritance was recently discovered in Plymouth rock chicken. Evidence supports rare instances of male mitochondrial inheritance in some mammals as well. Three-parent inheritance. An artificial reproductive process known as mitochondrial donation or three-parent in vitro fertilization results in offspring containing mitochondrial DNA from a donor female and nuclear DNA from another female and a male. In the spindle transfer procedure, the nucleus of an egg is inserted into the cytoplasm of an egg from a donor female which has had its nucleus removed but still contains the donor female's mitochondrial DNA. The composed egg is then fertilized with the male sperm. The procedure is used when a woman with genetically defective mitochondria wishes to procreate and produce offspring with healthy mitochondria. The first known child to be born as a result of spindle transfer (TPIVF) was a boy born to a Jordanian couple in Mexico on April 6, 2016. With this, we conclude today's lecture. Hope you have understood and enjoyed well. Thank you and goodbye.